Tails are destroyed so often, but using a nail clipper, they grow back. Made of the protein keratin, nails offer us protection from damage. When an infant's fingertip is amputated, but only the part beyond the last joint, it grows back to its original shape. A study published in the year 2013 by Takeo and colleagues found that there are stem cells in the nail bed. These are cells that can develop into different cell types. They do not only continuously differentiate into cells that go on to form the hard, keratinized nail plate, but they also send signals that orchestrate digit regeneration. These cells activate the WNT signaling pathway, a process that attracts the nerves needed for regeneration. The lizard is an unwanted guest in our homes. We often try to kill it, but often succeed in only hitting its tail. The fugitive soon regenerates its tail, just like the injured fingertip mentioned above. The molecular biology of this process was summarized by Hutchins and colleagues in the year 2014. More than 300 genes are involved in the response process, and developmental and wound response pathways are activated to heal the wound and put the tail back. The researchers investigated how the root tip organ, with a tapered shape, reclaimed its shape when amputated. They discovered that the existing cells changed their geometry to guide the alignment of rows of new, regenerating cells along an inclined path. These inclined rows of cells finally rebuild the tapered shape of the root. In technical terms, this process is called morphogenesis, the biological process that causes an organism to develop its shape and structure. It involves the coordinated growth, division, differentiation, and organization of cells to form tissues, organs, and even a complete organism. Experiments described in this study used root tip regeneration in Arabidopsis thaliana of the mustard family as an experimental system to demonstrate how changes in cell geometry contribute to and facilitate morphogenesis. Using advanced microscopy and experimental tools, the team observed that normal cube-like root cells changed their size and shape into rhomboid shapes, where the altered cells divided diagonally, producing triangular prism-like cells. The diagonal divisions redirected the growth of neighboring cells along a slanted path, collectively recreating the lost tapered tip. Mathematical and physical analysis done by the group and their collaborators revealed that the actual force behind these changes is internal mechanical tension, hidden stress guiding cells to change shape and divide strategically. Thus, while stem cells and genetic pathways have long been central to understanding morphogenesis, this study highlights an equally important but often overlooked aspect: how cells adapt their shape under stress. It shows that cell size and geometry can play a decisive role in guiding morphogenesis during tissue regeneration. These principles may even hold true across other biological systems, pointing to a more universal role for cell physical cues, in particular cell geometry in shaping life. Credits: script based on research by Takeo et al., Nature, 2013, and Hutchins et al., PLOS 1, 2014.